Hello, everyone, and welcome to today's webinar. I'm Charlene Lancaster, Assistant Science Editor for The Scientist, and I'll be moderating our discussion. Today, our speaker, Felix Lansing, will outline how AI-driven models are accelerating recombinase development, offering scalable and programmable tools to expand the therapeutic potential of gene editing. Before we begin, I'd like to thank Twist Bioscience for sponsoring today's webinar. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point, and our speaker will address these during the Q&A session following the presentation. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. Our sponsor has provided us with some helpful resources related to today's webinar, and we've posted these in our handout section on the right side of your screen. You can access and download these documents at any time during the webinar. If you've minimized the Q&A panel, simply click on the blue chat button in the bottom right corner to bring it back. This webinar will be archived on the Scientist website, and we will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. And with that, let me introduce our speaker. Dr. Felix Lansing is a visionary scientist and entrepreneur who co-founded SeamlessTx, a biotech startup focused on advancing gene therapies for hereditary diseases using programmable recombinases. In 2017, he embarked on a pioneering PhD thesis at Dresden University of Technology, where he developed programmable recombinases for hemophilia A treatment. The research yielded groundbreaking results, earning him the highest distinction and leading to several patent applications, publications, and awards. Now, as the Chief Technology Officer at SeamlessTx, Lansing leads a dedicated team revolutionizing gene editing by providing effective and accessible gene therapies for hereditary diseases and other severe disorders based on programmable recombinases. Let's make sure your slides are up and running. Okay, Felix, take it away.
Yeah, thank you very much, and thank you for the kind introduction. My name is Felix Lansing. I'm co-founder and CTO of SeamlessTx, and I'd like to share some insights on how we deploy machine learning on accelerating recombinase programming. So why are we working with recombinases in the sense of using them for therapy? If you look at the landscape of human disease, you will find that it's very immense. And if you have a closer look, you will find that many diseases are caused by mutations in our genome. And while there are many tools available to treat genetic diseases, it still remains very challenging to perform large edits. And here our platform comes into place that takes recombinases that are specifically known to perform large edits, and we can use the technology that we have developed to program them to direct unique sites in the human genome. Another feature of recombinase is that they are super precise. What does it mean? It means that the recombination reaction itself happens without any cofactors from the cell. Therefore, we are 100% sure about the edit, and we can always predict to the nucleotide how the recombinase will modify the genome. And as mentioned, large edits still remains challenging. Either deleting or integrating large DNA fragments from the human genome could, though, offer several curative approaches for many diseases. So we envision using targeted deletions or integrations as a potential curative solution for many genetic diseases. So looking at these two reactions I depicted here as schematics is first the targeted integration. What you need is a target site here depicted in blue in the genome and a partnering target site on the DNA donor template. A single recombinase can then form a homotetrameric complex, recombining these two target sites in this tetramer of recombinases. And when the recombination reaction is being performed, it actually integrates the DNA donor at a given sequence. And it has been shown by us and by others that you can really integrate large payloads with high efficiencies in vitro, but also people are moving this technology already now, similar to us, to in vivo applications. On the other hand, you have targeted deletion. In this case, you have two matching DNA sequences, the target sites here depicted in green, on the same DNA strand. And when the tetramer forms, it will basically excise whatever is in between those two target sites. And it will remain with one of the target site being left in the genome, and the rest will be excited. And you might know the Krelog-SP system that has been used for decades in basic research to, for example, perform conditional knockout using exactly this mechanism of action. So what is the challenge using recombinases for therapies? Basically, recombinases recognize a sequence, the wild-type recognition sequence. And even though they have been discovered in the 80s, since now there's no recombinase in the clinic. And this comes through the fact that these sites the recombinase are recognizing are not present in the human genome. And especially they're not present in any location that is useful for therapies. So they have been used in basic research where you can take, for example, an animal model and integrate the wild-type target sites at a given choice and then apply the recombinase to perform the edit. What we want to do at Seamless is taking the recombinase and programming them directly to a site found naturally in the human genome. And our first evaluation of the human genome shows us that there are potentially 15 million unique sites that we can target with our technology. And our vision is really to develop programmable recombinases as therapies to really treat genetic diseases, specifically focusing on large edits. So how do we do that? It all starts with the target site selection. So we look into the human genome for target sites that are suitable for our programming technology. That said, we have data on every single nucleotide of the recognition site, how difficult it is to engineer the recombinase for that given nucleotide. So we basically scan the human genome with our search motive, and we will define in a given locus many recombinase target site, where we then start the recombinase programming. The programming starts with selecting the starting library. What does it mean? So basically we are using directed evolution, or in other words, breeding of the recombinases. And before starting that process, we assemble a library of different recombinases that we have already generated. And these are in our freezers here in Dresden and in the US. We basically take the starting library, mix different families of already evolved recombinases to then fuel that in the reprogramming, what you see on the top. And the reprogramming is a process of positive negative selection over really rounds of generations, which we call cycles of directed evolution, where we perform selection, mutagenesis, and generational cycles to breed these enzymes or to program these enzymes against the target sites that we have selected in the first place. The result of the programming is a library of 20,000 or more variants that are recognizing their target sequence. So we also developed screening methodologies to look at multiple thousands of clones at once on different on and off targets to then really boil it down to a few candidates that we push forward for the in vitro and in vivo validation. If activity or specificity needs to be increased at this step, we also have several engineering approaches on working on the recombinase post-evolution. So how do we want to use machine learning to accelerate that process? Because it's very time intense and tedious. So we have different avenues. One is the target site selection. So we use machine learning to basically dig in our libraries and extract the properties of each library and how they recognize the target site. Thereby we can develop position weight matrices that can be used for searching the human genome, and then our model evaluates the evolvability of a given site in the human genome that we can then use to rank order our target sites to already have a higher success rate of evolving the recombinase against that chosen target. Another way of using AI is in the programming step. So as I have mentioned, we perform these rounds of iterative cycles of directed evolution. And obviously, they are based on random mutagenesis and selection factors. And what we can do is with AI to basically predict changes within the programming step that would be beneficial and speed up the program. And I'm going to share some more data on this particular step. And last but not least, also in the lead optimization, where we can use our models to basically take an existing library and we task the models to basically predict better variants that are not present in that library, but should perform better than the best clones derived from evolution. So you see there's several avenues on how we can deploy machine learning to accelerate the discovery of recombinases. So as mentioned, I want to focus today's talk on how we basically improve properties of evolved recombinases. Basically, by deploying machine learning to either enhance activity on the on-target or to decrease activity on potential off-targets. So let's come to the material and data set that we have available at SeamlessTx. As I mentioned, we have performed directed evolution already for hundreds of different targets in the human genome. And this derives to recombinase libraries that we have banked as physical materials in our freezers. So by to date, we have more than 10,000 recombinase libraries that have been evolved for different therapeutic targets. And each of these libraries contains more than 20,000 recombinase variants. So this gives us at hand approximately 200 million recombinase variants that we have physically present as material in the freezers. What we're doing now and investing in is sequencing these libraries, and we are going to sequence them based on the profiling where we connect the amino acid sequence of a given recombinase to the activity on a given target site. And it doesn't need to be only the on-target. We can profile these recombinases on several multiple target sites. So you can imagine we have in-depth data on each amino acid sequence of all of these clones on their activity on a given DNA target. And this is basically the foundation of our machine learning model training. So how does the data set look like? We basically equip the recombinase with a barcode. Then we task them to recombine their targets. And once they have recombined their targets, we linearize all these plasmids. We sequence then the recombinase and the target site, either recombined and non-recombined. And then we can basically calculate recombination efficiency based on sequencing for each of the clones in this library. And the data sets look usually like this. Here we sequence a given library on 3 different targets. You see them on the y-axis and the x-axis, and color-coded also the third target of the dots. Each dot is a single recombinase variant, and you now have data on the amino acid sequence of, for example, this clone on all 3 targets. So we know that amino acid sequence of that clone gives us activity X on target 1, activity Y on target 2, and activity Z on target 3. So you can really now group these recombinases and cluster them depending on the activity on the targets, etc. And this is the typical data set we get from a single library when we, for example, now sequence it for 3 different targets. So this is the base of our machine learning approaches. We have 2 components actually that are used to predict recombinase variants with better properties. The first one is the so-called oracle. And the oracle basically takes the protein sequence input and predicts an activity of that given protein sequence. So the protein sequence obviously the recombinase, and what it does, it basically tells us, okay, if we have that sequence, the recombinase will have a given activity on a DNA target. And we train this model obviously with the sequencing data that I just shared. So we fuel it with thousands of different data points on amino acid sequences correlated to their respective activity on the DNA target. So this is our model to evaluate and to predict activities of a given sequence. However, you need new sequences to predict activities and potentially create better variants. So the second component of our model is the protein generator. Again, this takes into account an evolution library sequencing data, and it basically performs in silico evolution. So it takes the fittest variants, so the top variants with the highest activity on a given target site, and then performs mutagenesis on these and creates new amino acid sequences. The mutagenesis is biased based on all the mutations we find in our library, and also if you change the different parameters, you can allow new mutations that we have not found yet using directed molecular evolution. So it basically evolves the recombinase by in silico predicting new variants, and then our oracle is predicting the activities, and these then go back into the protein generation tool, and we basically perform this whole process thousands of rounds, mimicking evolution in silico with these 2 modules. And at the end of the modeling, we collect the most active variants for any of the stages of the iterative process of these two modules. And these candidates, we nominate them for validation. So how does our pipeline look like on discovering recombinases? So here you see our evolution. We make recombinase for a given new target. We then perform the sequencing. And once we have the data set, we basically pick the top performers and we nominate them as natural variants from the library to be validated in vitro, either in bacteria or human cells. Next to it, we are then training our model. We split our data sets into fractions, a test and a train set. We then train our models with the train set. We leave out the test set, and then we evaluate the accuracy of our model with the test set. And then we have, let's say, the model that passes our QC will then be allowed to generate optimized candidates that then again fuel into the testing. And here we are reliant on a quick synthesis process of DNA fragments at scale with the exact sequence that our model is predicting. And here we're using the Twist fragment synthesis services, where we really have a very quick process of synthesizing these fragments and fueling them in our vitro test validation loop. So I want to share how a typical data set looks like. What you see here on the left plot is the AI model performance. So again, as mentioned in the two colors, we have the training set in light green and the validation set in blue. And we have the activity that is being measured. That's the real data coming from the sequencing. And then we have the predicted activity from the oracle. And what we want to have is a strong correlation of these two parameters, because then we can say, okay, this model passes our QC and is allowed to predict beta variants. And what you see on the top left, it's the predicted beta variants. Obviously, they have no activity measured because they're in silico generated, and we don't have activity data set on those yet. And if you plot these in a histogram, you will see that here again, you see all of our data, real data of recombinases from our sequencing. In dark blue is the top 12.5% of the data set plotted. And what you can see here is that our oracle evaluates the newly predicted variants as being better. Obviously, that's all theory, so we need to test these predictions and validate them against our best candidates from the evolution library. So when ordering these fragments, we first perform a QC by sequencing, because we want to make sure that we have each of the predicted fragments and recombinases present as we designed them. Because using recombinase, you might know that a single amino acid changes in one of the conserved regions can really destroy the whole activity of the enzyme. So we rely on high accuracy and low error rate during the synthesis process. And that's what we were checking for. So we see here is 3 examples of library 1, 2, and 3. Histogram again showing sequencing reads, so we have equal distribution nearly of the variants inside the library, and also usually 95 to 99% of what we sequence is matching actually what we were expecting. So we have a high accuracy, and also the error rate is pretty low. So most of the fragments that we are synthesizing match our QC to then be moved forward for the testing. So here's 3 examples on how the data looks like. You again see the real clones from the evolution library called library selected clones, and then our predicted variants that should perform better. And this is 3 examples how the data can look like. We normalize it always to the most active natural variant, so to the one that is coming straight away from evolution. And in some cases, we predict variants that are just slightly better. Here, the best ones are performing 6% better than the natural occurring. In other cases, we can get activity increases of 30 to 50%, just showcasing here one where we had 35% increased activity of the predicted variants. And in also other cases, depending on also how depth you go into the data set, you can predict variants that are 3, 4, 5-fold better than the best natural clone. And you can imagine that evolution is able to gradually provide you with better variants, but having a jump of a fold change is very hard to achieve with directed evolution. And here we can really accelerate the process of making these variants quicker and better using machine learning. So Coming to the end of the talk, I want to share my vision on how we use and integrate machine learning, the program of Recombinase, and the quick turnaround time of synthesizing fragments from Twist and incorporate them in our workflow. So I'll give you two examples, one on the programming and one on the lead optimization, how I see that we deploy this to create enzymes with higher speed. So as I mentioned, the programming takes usually 60 to 80 cycles of directed evolution. What I'm imagining is that you now start evolving an enzyme for a given target site. And after 5-10 cycles of evolution, you basically start sequencing that library. You use our models to predict variants that are better performing than what has been generated by directed evolution. And the quick turnaround time, you can synthesize 500,000 fragments that are being predicted to be better. And they will be incorporated in the evolution with family shuffling, for example, that you now have natural mutations and randomly occurring mutations coupled with machine learning aided prediction of better variants. And we can mix the two, and thereby basically you are increasing the diversity of your evolution library. And as I said, you might have some variants inside that are a couple of fold more active than the evolved variants, thereby speeding up the process of the programming. On the other hand, obviously, when we are done with evolution, we're aiming for therapeutic editing efficiency. And if, for example, evolution library natural variants are not at these efficiencies, we can use, as showcased also in the examples on previous slides, the machine learning and the synthesis to generate recombinases and recombinase libraries that might be better than what natural evolution has been delivering. And obviously, what is also important is that we always fuel back this data into our models, because the models just get better with data, and the more data we have, the better the models will be. So that's also a self-amplifying process where we will only get better by generating more data to refine our models, to refine our parameters, and to then eventually be able to really accelerate the development of programmable recombinases. So I hope I could convince you that using machine learning, also synthetic biology by synthesizing fragments and deploying them in your workflows might accelerate processes that are otherwise tedious and time-consuming. And with this, I'm happy to take questions. Thank you.
High-throughput screening is fundamental to biomedical research, particularly for the development of protein-based therapeutics. By iteratively screening protein variants, researchers explore a protein's vast sequence space in search of a novel variant with desired, optimized properties. However, finding the right protein variant is difficult, even with the latest AI tools. Large-scale screens and training datasets for machine learning require pools of DNA, encoding thousands of defined protein variants to effectively sample the protein sequence space. There are many ways that researchers can generate their screening libraries, but current technologies leave much of the sequence space out of reach and underexplored. If your protein is longer than 300 nucleotides, your options for building screens are either very expensive, very limited, or very biased. As a result, the sequence space of most known protein sequences cannot be effectively studied in high throughput. To advance therapeutic protein development and AI-driven screening, researchers need tools to economically synthesize longer sequences at scale. At Twist, we constantly ask, how can we give more scientists access to longer synthetic DNA on a large scale? Our team used our proprietary silicon-based platform, optimized chemistry and automation to become the only company able to synthesize precisely designed pooled sequences that push the boundary beyond 300 nucleotides. This innovative new product, called Twist Gene Fragments, consists of uniform pools of double-stranded DNA that are designed to your sequence specifications. In just 1 week, you can receive pooled lyophilized gene fragments, up to 500 base pairs ready for use in your high-throughput screen. With access to significantly more sequence space, you could precisely synthesize a library to generate training datasets to improve your machine learning model, or screen single-domain heavy-chain antibody variable regions and make discoveries that will lead to the next blockbuster protein therapeutic. So, what will you do with significantly more sequence space? Get started with multiplex gene fragments today.
Thank you, Felix, for the presentation. So the audience has already submitted several questions, so let's get to them. So what diseases is Seamless Therapeutics focusing on?
We have not yet disclosed on what diseases we are working on, but as I mentioned in the beginning of the talk, we are focusing on large edits. So we are looking at diseases where a large deletion or large insertion can be a curative approach. And additionally, what I can share is that we would like to use single recombinase also to be applicable on several diseases. So if you take the example of DNA integration, you can think of two modalities, either using a gene of interest with an endogenous promoter where you integrate an intronic region to then use the endogenous regulation of the gene for expressing your therapeutic payload, or you can use a safe harbor region where you bring in your own promoter, maybe a tissue-specific promoter, to then express your cargo of choice. So we're currently evaluating different targets and based on efficiencies, and we will decide what will be our lead program, but it's not disclosed yet.
Okay. And what led the company to start leveraging AI? Was that always in the plan when it initially formed?
I have to say it was in the plan. We have a strong focus on developing a therapy, so bringing something to the clinic. And we have seen on the way that we have so much data. So we're evolving recombinases already in the academic leg before the spin-out and also now at large scale at Seamless. And what we have realized is that we have just this immense data set of physically present libraries in the freezers and their activity on different target sites. So we were in a publication in 2022, we saw first evidence that you can use machine learning to predict variants that are either better or, let's say, in a further stage of evolution. And that led us really to leverage now the machine learning for our purposes. One bottleneck that we have been facing in the beginning was actually the synthesis cost plus times that didn't allow us to integrate that in our evolution process. So recombinases are 1,000 to 1,500 base pairs long, and it is really hard to get exactly these sequences at scale to in a quick time that you then can fuel in and that gives you a benefit. So also I think the progress made in synthetic biology, also reduced cost in synthesis, etc., allowed us now to incorporate that really at scale. So I think these two factors made it now possible to incorporate it.
Great. And kind of what are your next steps or your company's next steps in the next coming years? What should we expect from you?
So as I said, we are heavily focusing on developing a therapy. That's our first priority, getting the recombinase to the clinic. Today, no recombinase has been used for treating a genetic disease. Other modalities obviously have moved in the clinic and are also first therapies being approved. And secondary to that, we want to offer really programmable recombinase as a tool for the broader range of potential users, including others that want to make therapies out of those, but also eventually at a later stage at low cost providing tailored recombinase for any kind of questions. These enzymes have been used in the past for basic research, but they were limited to the few wild type enzymes. And if you could extend this now with reprogrammed recombinase, I also see a path of really being the company providing recombinase for any kind of application.
And are you collaborating with researchers that are also interested in using recombinases?
So at this stage, as I mentioned, it's still pretty cost intense and tedious to make recombinases. So we're currently focusing on our internal development. Once we are getting better, also we're counting on our machine learning to improve and accelerate this process. We will be open also to collaborate with any kind of interested folks. So this stage, I would say it's not at the stage where we could deliver at large scale for everyone a recombinase, but in the future, that's definitely something we are looking at.
Great. And so once the lead candidate is selected, is there any experiments that you perform to determine the accuracy?
Yeah, so I think we are following different modalities on checking on the precision of our editing tools. We have non-biased whole genome sequencing as one of the methods, specifically looking at large rearrangements. Then we obviously have bioinformatically predicted sites that we are checking in a synthetic fashion. For example, we have a methodology where we can take a single recombinase and get high accuracy activity data, more than 6,000 potential off targets. And then also we are looking at general properties, long-term healthiness of the cells, etc., and assessing also other effects of the recombinase. So we're looking at these three factors, obviously, because when providing a therapy needs to be safe and it should not cause any other alterations of the genome than the expected edit.
Sure. And I think along those lines, is there a risk of recombinases causing rearrangements between homologous chromosomes?
Yeah, so I think it has been shown also that this can happen at high level of recombinase expression. There are several labs that have looked into it. However, it is not likely to happen if you have a high precise on-target editing. And also what we have been showing to avoid this is that using our recombinase engineering approaches, we published it last year, that we could integrate another DNA binding motif like a zinc finger into the recombinase as a safety switch. So what we have shown is that only if that zinc finger is binding a sequence next to the on-target, the recombinase becomes active and we can recombine the target where we have reprogrammed before. So we are establishing also safety switches by engineering these enzymes beyond evolving them to provide a safe module for gene therapy.
Wonderful. Well, thank you, Felix, for that discussion. We appreciate your presence here today.
Thank you very much, and thank you for inviting me.
Wonderful. If anyone has further questions, please consider reaching out to the speaker directly. His contact is shown on the screen. As a reminder, the webinar will be archived on the scientist website, and you'll receive an email notifying you when the on-demand webinar is available. Make sure to follow the scientists on social media for updates on future webinars. I'd like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientists, I'd also like to thank our speaker, Dr. Felix Lansing, as well as our sponsor, Twist Bioscience. Thanks, everyone, and goodbye.